Right. So I hope that you now realize the importance of contrast. So we have the optical resolution, but if we have no contrast, we're not going to see anything in the image, or we cannot analyze at all. And also of digitalization, so sampling. The way we acquire the images also matters and can limit the resolution in the image. And what happens if we do not sample correctly? I said we are creating artifacts in the image. So this is what we're going to look at right now. Just want to click. Right. So these aliasing artifacts are very, very well known in the world of photography or medical imaging or in astrology, but for some reason, they are forgotten in biology. So we need to put them back into our brain and see what happens. So we have some artifacts are visible. So this is the reason why it's well known in photography and in medical imaging. It's because it is visible then, and that is especially true if you have colors. So that's why photographs really bother about that. So you can see here pixelation. So this is due to the fact that we are measuring our optical image in discrete points. So when we have a line, we get, instead of having a smooth line, we get a jagged line like this. This is pixelation, so it's due to digitalization. And then we get moire as well. Moire is, as you can see in here, this is due to undersampling. So moire is a pattern or a color that are created in the image and that do not exist normally. So this one is sampled properly, has no moire patterns, but it anyway has pixelation because we have to, we have basically discrete points. So, but the moire patterns disappear when you do not undersample, when you sample properly. Unfortunately, there are other artifacts that are not visible, but the information in the undersampled image is incorrect. So we always need to have the right balance between, is it okay? Will this incorrect information prevent me from getting reliable answer for my scientific question or not? That is the key. It's always the key, okay? So moire is a subtype of aliasing artifacts, and they are very easy to see. You can see something like, if you take a picture of your screen, you will see things like this on the image. And these are moire patterns. Once you start seeing them, you see them everywhere. For example, if you have a fabric, like a thin curtain in front of the light, sometimes you see these moire patterns in the curtain. So once you see them all over the place. This happens, moire patterns happen when we have two superimposed structures. So in our case, we have a structure in the sample, and we have the camera chip, which is a structured, it has a grid. So what you can see in here, if you superimpose two structures, is that you get these black lines that are thick, that are created, and do not exist in the sample. And so the moire adds to the pattern some things that do not exist adds to the image some patterns that do not exist in the sample. And this is orientation dependent. This is the reason in here we have all these wavy lines instead of having straight, thick lines like this. It's because it depends on the relative orientation of the two structures. So if one structure is not completely flat, like a screen is a bit not completely flat, then you can see that it changes with the orientation. This does not happen if we image at Nyquist sampling, so when the sampling is more than 2. Moire is very rare in images of biological samples because we have no regular patterns in our samples, and that's the reason why we do not get moire. So I recommend really to watch this very nice video again by Jeff Lichtman in the iBiology microscopy course. I have the link in here where he talks about sampling. So in these images, I took some screenshots, and in these images, you will see we are using always the same objective and the same object. And I guess quite quickly you will recognize what this object is, but we're increasing the sampling. So you can see this poor zebra. I'm sure you have guessed it is a zebra. And we are now imaging the zebra with a very low sampling, so very big pixels. And what we can see is we see things that actually do not exist. It's not possible. The zebras do not have bridges like this. So some things are created in the image because of undersampling. Okay. So if we sample a little bit better, what we can see now is that, oh, this bridge disappears. But what we can see in here now, we have this sort of checkered pattern. Again, this is not something the zebras have. So again, we have things that are created here, bridges uh, uh, between the objects. And if we look here in the background, we have this sort of the poor zebra is not in the jungle, so or in the desert. So is we have all these balls like this in the fence behind. If we now sample a little bit better, now we can see that there is no bridges between the black lines. We have lost the this checkered pattern that we have here, but the poor guy got a hairdo, a frizzy hair, which zebras do not have. Okay, so this is again an artifact. And we can see in here also that we have all sorts of strange pattern. And now when we see a little bit better the fence behind, but we shouldn't have these little balls everywhere. So now we keep on going and we are sampling even better. So we start getting some nice definition of the zebra, but it still gets frizzy hair. And here we have a very strange pattern. Okay, slowly again, here is an even better digital resolution, but we still get these very fine details. You can see that what's left as we go with better and better digitalization is the fine details are still not digitally resolved. So the other details, so everything in here is resolved by the objective, but the other details are now well resolved, but only the finest details are not digitally resolved. Okay, so we keep on going and now we are resolving even the fine lines on the nose of the zebra. So we compare an image that is undersampled with an image that is sampled. You can see that there's a lot of things here in this image that are wrong, like seriously wrong. Okay. The problem is that we do not know what our samples look like. So the information in the undersample images is incorrect. It's not possible to detect because we do not know what to compare with. We don't know what the image should look like. So depending on the scientific question, undersampling can lead to dramatic interpretation errors. And I cannot stress enough, depending on the scientific questions, right? So this is what we always come back to. So undersampling creates aliasing artifacts, and we rarely see them, unfortunately. So what about a sample without a regular pattern? Because the zebra is anyway has regular patterns, right? So we could see the aliasing. Well, it's even worse because we cannot detect them. So now I want to show you this random line sample. So this is something that you can get from TIFF and it creates from in Fiji, and it creates random lines. Okay. If now, so I have this line pattern and it's a pattern, but it's no regular pattern. And I'm going to zoom the image in and out, and that is, of course, overlaid with the screen pattern. The screen has a pattern, right? So when I zoom into the image, what I can see is that I have some digitalization artifacts. So these lines are jagged because we are taking discrete samples. And we also see at the places where the lines cross that we have this big black blob, which normally we shouldn't have. But this big black blob is because we cannot resolve these. Okay, so that's what we are seeing. And in here, it's the digitalization artifact. But now if I zoom out, look what happens. Where are the lines? The more I zoom out, and the basically I still have the same resolution, digital resolution in the background in my camera or my screen, but I make the objects finer and finer. And you can see what happens. I'm losing the lines. Now I'm getting lots of little dots. But you see, this line is still there. Somewhere in here, there is a line like this. Yeah, this one is still there. And this one is still there. So when I zoom in and out, some of the lines are correctly displayed, and then the rest is all scrambled. So it is it depends. This is what I'm saying, that it depends on the orientation. So the relative orientation of the screen versus these lines, they can be sometimes well digitalized and sometimes not. And the problem is worse with low magnifications. So again, matching the pixel size with the, in this case, the screen size or the camera size when we image up with a camera, then the problem is worse with low magnifications. So the lines look different depending on their orientations compared to the screen or the camera. So this is the screenshot of random lines zoom in and out on the screen and with a fixed size like a camera. So I had the luck of having a great discussion with Rainer Heinzmann, who said immediately, I'm going to model it for you. And so he did. So I was telling him, okay, but I was puzzled, is like in our images, in our samples, we have no pattern, no regular pattern. So does it matter if we undersample? Because in biology, we undersample a lot. If you listen to any physicist, they're going to tell you never undersample because the information is wrong. So in here, you can see he created all these objects with different orientations and different frequencies, right? Because physicists love frequencies. And in this one, we have a very fine object with a little dot in the middle, a little black dot in the middle. And then we have all this. And in here, we have some objects that look a little bit more like a biological sample. So in here, we have some random pattern of dots, okay? If we undersample 3 times, what do we see? And this is a camera-based system. We see that we get more artifacts that are visible. And then in here, everything is fused, right? Even though it was resolved, it is fused. And then we can get here, we're lost the resolution. So we are not resolving the optical resolution. The digital resolution is too low. And then you can see in here some strange stuff happening compared to here. So things are fused and they make bizarre bridges. And you can see in here as well, here we have pixelation. This is the digitalization process. But if we look at the stuff that looks a little bit closer to our microscopy sample, what can you see? You can see in here that these 3 dots that were resolved are actually not resolved anymore, and they make like a little banana like this. But we can see in here that we also have something that looks like it's elongated, whereas it was just a simple dot. All these dots have the same intensity and the same size to start with. And if we look in here, we have another strange banana, and one of these dots look much dimmer than the others, even though in here it looks the same, and we resolve them. And we can look at these 2 dots here. They have the same size and the same intensity, but one of them looks much dimmer than the other in this undersampled image. Okay? So now if we're looking at what happens in a single point confocal instead, it's the same thing basically. You can see pretty much the same type of artifacts in this undersampled image in a single point confocal. So on a single point confocal, the situation is a little bit different though, because as you know, single point confocals are slow, and an inexperienced microscopist will want to use less pixels. Sorry. So if we have maybe, I don't want to image with 2048 pixels. Now I'm going to put 1024. It's good enough. This is in general what happens because people want to go faster. And then we use a great objective, right? But still, we undersample. So in camera-based systems, we have large pixels, but they are next to each other. So there is no space between the pixels where we do not collect light in a camera-based system. So in a camera-based system, because we have an array of pixels that cover the whole surface where the point spread function is projected. I'm sorry, I'm going to have to go and have a little drink of water. So just a second. Here I am again. So on the camera-based system, we're collecting all the light that comes from the optical image, one way or another. We can get bigger pixels or smaller pixels, but we collect everything. But that's not the case on a single point confocal. So when we undersample on a single point confocal, we create artifacts and we're missing data because we're jumping from one point to another point, and we're measuring here and we're measuring here. We're not measuring in between. And that is a big difference between a camera-based system and a single point confocal. We're actually jumping between spots, and whatever is between the spots, we're not measuring. So we can miss things. And we can create artifacts from this as well. So you can see here, for example, if I would have a sample that looks like these green lines, and now I'm scanning with my single point confocal, so I scan along these lines here, but I make big pixels because I want to go fast. So I jump from here to here without measuring anything in between. And so what happens is that I measure something here, and then I measure something here because there's some, it overlaps with my this green sample. And if you squint a little bit, what you're going to see is red lines, thick red lines in this direction. And this is what your image will look like. And anything that is here between the sampling point is not recorded. So you're going to be missing information. So the real signal, which is green, is not recorded. And the red thing is sort of makes a big thick line like this that we're going to see in the sample, even though it does not exist in the image. So thick red lines are detected, but they do not exist. And the green lines in the sample are not recorded between the sampling points. So, and you will see that in Z and in time, it's the same thing exactly as the single point confocal. But in the single point confocal, it's also the case in XY that if we have big pixels, we miss information. So we have to figure out what pixel size do we need. So this was for XY. So we have a difference between camera and the single point confocals. So what happens in Z? It's the same thing. When we acquire Z stack, and that is the same for all microscopes, okay? So now we don't need to talk about single point confocal or camera-based system. All the same when we acquire a Z stack, the object moves or the sample moves. So either we move the objective or we move the sample to the Z stack. And so we have a certain Z step between the planes. So we're acquiring image here, then we move to a certain distance, and we acquire another image, we move to another system. So what you can see is that also we are not recording between the Z planes. We're only recording at the Z planes. The great stuff with the Z stack is we have a magic button, and it's called Nyquist, or maybe it's called Optimal, or maybe it's called Recommended Z step, something like this. We have this magic button in the software, and that regardless of whether we are imaging with a single point confocal or a camera-based system, we have a magic button. But the Nyquist sampling theorem, what does that say? The smallest distance resolved in Z by the objective, so the optical resolution, must be sampled more than twice. So we need to have, so we know the optical resolution in Z, it's like this, and this is the radius of this big blob. And then the Z step that fulfills the Nyquist theorem is half of that, so we can just remove the 2 here, or smaller steps, okay? So we can make lots of little steps, but if we have a Z step that is bigger than that, then we are undersampling. So when scrolling through a Z stack, how do we choose the Z step that works for us? We have to think of what is the significant, well, related to the scientific question, what is the significant feature in my image that I want to record reliably? This feature should be visible in more than 2 Z planes for you to sample correctly. So here is a correctly sampled it's a nucleus with dots inside. It's correctly sampled. So if you look at any of the dots, you will see that they appear in several images, more than 2 images, okay? So what I want here is to count the number of dots. I don't want to miss any. So I don't want to have 2 big steps, otherwise I'm going to miss them because I will not record between the Z steps. So each of these dots is in more than 2 images, and therefore it is correctly sampled. If I now acquire with big Z, that's what I'm seeing. So for each z-plane, I'm going to see different dots. And it's very easy for me to miss some dots in between. Okay, so when you want to decide in all dimension, in x, y, in z, in time, when you have to decide first, what is it that I want to not miss? And that depends on your scientific question. And then you say, okay, what distance or what time does this correspond to? And you divide by 2. Okay, this is the way you decide how to sample things in the correct way. So this signal is not recorded between the z-planes. So now we have a little quiz. What are the image pixel size in XY then, and the Z-step or Z-stack that fulfill the Nyquist sampling? We are now, so I remind you the how to calculate the resolution in X, Y, and in Z. And then a Nyquist sampling is the size of the image pixels, sorry about that, divided by the Z-step. So you should be half of the optical resolution or smaller. So I would like you to tell me for a 60x numerical aperture 1.49, and this is an oil objective, so this is the refractive index of the oil, or for this objective, what is the XY optical resolution? You can calculate for a 60x. Anyone has the XY resolution? You've done this many times. The XY resolution of an objective. And yeah, I forgot to say, this is we're imaging EGFP, so it's 520 nanometers.
$212.
Okay, everybody agrees with that?
Yes.
Thank you, Adrian. Here we go, 213. I probably rounded up in a different way.
Yeah, because it was 212.8.
Yeah,
Right.
0.9.
Okay, good. And very good. And then if we would calculate for this one, we would see that it's worse. This is an air objective and it has a lower numerical aperture. So clearly it's going to have a little bit worse resolution. And now, oh yeah, what is the pixel size? I should have shown you, but anyway. How do we get the Nyquist image pixel size from that? Or how do we get, I should reformulate that, how do we get the size of the image pixels that fulfill the Nyquist criterion? What do we do once we have the optical resolution?
So it should be half of the resolution. So
Yes.
213 divided by 2,
Super
right?
simple. Yes, absolutely. So if once we have the optical resolution, we just divide by 2. Ta-da! And what does that mean? We would have, it should be 106 or smaller, right? Because if we have smaller pixels, we have more pixels and we have a higher digital resolution. So we definitely will resolve this. Okay, so it should be 106 or smaller. And in here, same thing, it's just half. All right, so what now is the Z optical resolution for this objective?
$712.
712. You guys are okay? Yes, bingo. And this is the Z resolution for this objective. Now, what is the Nyquist Z step? So what is the Z step when I do a Z stack? How much do I move between the steps in such a way that my Z stack fulfills the Nyquist criterion? and which means that I am sure that I am resolving what the objective gives me. And my Z-stack resolution is not going to be limited by my digitalization. So what would you
Does
say?
does the same apply here, like half of the set optical resolution?
Same thing. Super simple. I can tell you Nyquist works for everything. So it's very, very simple. Just divide by 2. And this is the set step that you should use, or smaller. So if you use lots of Z, you will also resolve this, but you should not use bigger step than this. Okay, this is the sort of the biggest step that you could use. You can use smaller as well. Okay, and it's the same thing. You see, it's very, very simple. We just want to make sure that the optical resolution is digitalized in the correct way. So we do not create artifacts when you do that. And we do not lose any resolution that the objective is giving us. So on a single point confocal, how can we get the image? What do we do on a single point confocal to fulfill the Nyquist theorem? Anybody can tell me?
We click on the magic button or
Yes. We click on the magic button. Super nice. You want to be to fulfill the Nyquist theorem and make sure that you collect everything that your objective gives you. Click on the Nyquist button. This is what happens on a single point confocal. And then what can you do with a system with a camera to make sure? There's no magic button, unfortunately. Not in XY.
magnification.
Yes. So we look in the software. We can see the image pixel size. We can always find, once we acquire an image, we can see somewhere in the software what the pixel size is in the image. And all we need to do is check that this pixel size is smaller than half of the optical resolution. Okay? So the pixel size in the image correspond to the, we can also calculate them. They are the size of the camera pixels in nanometers divided by the magnification. So it shows in the software, it's written in the software. All you need to do is check that the image pixel size is smaller than half of the optical resolutions. At the end of the day, it's pretty simple. Okay, what about on the Z-stack? What can we do to make sure that we are collecting everything that the objective is giving us when we acquire Z-stack? How do we set the Z step?
We can use the optimal step.
Yes,
recommended.
magic button.
Mm.
Okay, that's great. So we have a magic button for Z, and that is true for all types of microscopes, because acquiring a Z stack is exactly the same in all microscope. We're just moving the sample, or we're moving the stage. We're moving the sample, or we're moving the objective. So we acquire a Z stack in the same way on all type of microscope. We have a magic button. And this, again, is if we need to collect all the information that the objective is giving us, right? If our scientific question requires this. Robin, you had a question.
Does this the Z stepping take into consideration what the area unit the confocal is set to, or is there an easy way to, let's say you have an area unit of 1, how you then adjust this for the Z stepping?
Yes, absolutely. Because we know that if we close the pinhole smaller than 1 area unit, we're actually increasing the resolution in XY. And yes, I think that it does take in account. So the calculation is more complex, let's say, than what we have used in this course. But the I'm not sure, to tell you the truth. I don't know how they calculate it in the different software, but I would assume that they are doing their calculation properly, so that if you click on the Nyquist button, you get the correct Z step. I think that would be quite interesting to check. Maybe you can do when you go to your microscope, because you are closing down more than 1. So basically, the idea would be if you you set everything. So you set your zoom, the number of pixels. You click on the magic button. One of these two will change to go to the normal, the Nyquist pixel size, and that's in XY. But then you click on the magic button for pinhole 1, and you close the pinhole more than 1, so 0.5 or something like that, click on the magic button again, and we see if the Z step changes. And if you can, you let us know.
I'll check that.
It should change, right?
I did it the previous time, and yes, it changes.
Yeah, okay, good. It means that they are doing their job at doing the calculations. I was quite certain, but I've actually never tried myself. So if the point spread function becomes smaller, and maybe you have noticed this, if you change the wavelength on your macroscope, and if you change the objective, clearly, the 1 area unit is going to be different. So if you change objective on a single point confocal, and then you forget to click on the 1 area unit, you might end up imaging with a very small pinhole or a very big pinhole. So it's important when you change something that you click on the 1 area unit again. Okay, very good. So you see, it's not super complicated, but I think you understand that we need to digitalize the images in such a way that we're not creating artifacts, and also that we are collecting everything that the objective is giving us. And again, that depends on the scientific question. So we will look at this, but we always need to relate this to our scientific question. If our scientific question does not require a high resolution, if we have in X, Y, or Z, if we have very big differences between, for example, the control and so we're not after fine details in the differences between the control and the wild type or whatever you want. In that case, you can do whatever you want. But when you are trying to get small crowded objects, and you are trying to find small differences between your control and your treated sample, this is when you have to be very careful with this. So in time lapses, it's the same thing. So as I said, it works for everything. So we need to, in a time lapse, we need to define. So basically the question is, I want to do a time lapse and image my samples live and see how things change. So maybe the intensity, maybe the shape of the cells or something like this. How often do I image? How do I decide this? It depends on the scientific question. So you figure out, and you have to involve your knowledge of the sample biology, clearly. You have to figure out how long it takes for a significant change to happen. Considering your scientific question, what is a significant change and how long does it take? And when you know this, well, you just need to image more than twice in that time interval. So Nyquist works also in time. So you can see in here, we have calcium imaging of cardiomyocytes that are pulsing, and they are pulsing a calcium pulse. So the scientific question is, we're imaging for whatever, 20 minutes or something like this. It's a lot shorter than 20 minutes, this one. But let's say I want to image for 20 minutes. How many pulses do I have within these 20 minutes? That would be one of the scientific questions. And then the next scientific question is, for each of the pulses, where exactly is the peak? So how long does it take to peak, right? So these are our scientific questions, and now we're trying to figure out what interval do I have? How long time between my images? So if I have 20 millisecond interval, this is what I'm seeing. So I see that in this, what it looks like within my time lapse here, I have one pulse. Okay, that's what it looks like. And then if we look carefully, maybe we can define where the peak of the pulse is. So maybe we would say here is the peak of the pulse or something like this. So we're quite precise into knowing. But if now we decide to instead image at 200 millisecond interval, so 10 times less, this is what we're seeing. And now we need to ask ourselves, well, there is a significant change in the intensity between any two time points. There's a huge change in intensity. I am not sure if I have enough, if I'm imaging enough in here to see if there's one pulse or two pulses and where the peak is. But now, if we have some knowledge about the sample, and we know that the peak is longer than 400, right? If we know that the peak is longer than 400, then we know that 200 milliseconds is going to be sufficient, right? So you involve your knowledge of biology to know how long it takes for a significant change to happen when it comes to your scientific question. How long is a significant change? You divide this by 2. This is the longest time you can use. You can choose shorter, but then you're going to be maybe bleaching the sample and damaging the sample. But the minimum absolute biggest time you can use is going to be half of the significant change. Okay, so in this case, for example, it is possible that we oversample here. Maybe if we had, instead of 10 times longer interval, if we had double, like 40 milliseconds, maybe we would also be able to collect the same information. So we need to have some sort of knowledge of the sample to define the time interval. So the Nyquist theorem for time works as well. The time where a significant change occurs must be sampled more than twice. So 2 time points or more. Okay. So when are we okay with undersampling in this case? I'm not saying that everybody in here must match the refractive index of their sample with the objective. Totally not. It depends on your scientific question. I'm not saying that everybody has from now on needs to image Nyquist sampling. Absolutely not. There are some cases where it's okay to undersample. So let's have a look. Sometimes we're stuck. Light toxicity for live samples and some objective properties can force us to undersample. But what is important is that we know the consequences. And that for our, even if we undersample, as long as we can get reliable result for our scientific question, it's okay. So if you can see in here in these 2 images, this one is undersampled and this one is sampled correctly. So you can see in here some worry artifacts in here because this fabric is very fine details. So it is undersampled here. Whereas this part here is very coarse details in this image. And so it's properly sampled in here. So we have no artifacts in here. So only the details that are undersampled create aliasing artifacts, not the stuff that are correctly sampled. So you can see in here, coarse details, no artifacts. Fine details, we get more artifacts in here, okay? So the closer to Nyquist sampling we image, so half of the optical resolution, then the less are aliasing artifact and the more accurate the information in the image. But, and also the less information in missing, not recorded, if we consider single point confocal that jump between the spots, and also Z-stack and time. Okay, so we really, we need to consider this, that it's a continuum and all the details. So it's totally okay to acquire an image like this if you're not interested in this structure. If you're only interested in this structure, you're going to be fine with this undersampled image. So how good is good enough? The choice of objective and sampling must be driven by a well-formulated scientific question with metrics. What do I want to measure in this image? So quantifiable metrics of distance, volume, time, etc. that need to be resolved, and they must be correctly sampled. So considering our knowledge of the biology of the sample, how large are the expected variation between, for example, wild type and mutant or something like this? If you have small variations that you want to detect, you must be much more careful and sample everything that you need to extract in a Nyquist way. If you have big variations, you're okay with whatever. So the smaller the difference, the more accurate the analysis must be. And you must then be very careful with having as best a sample possible and excellent imaging. So for example, if you acquire an overview of your sample, what the scientific question that you have, if you want to say with this image, is where are the border of my sample? You do not need any high resolution for that. So you can do whatever you want. Use a low magnification, low NA objective. Make enormous pixels, fine. As long as it shows you the border, you are reliably answering your scientific question. Okay? So we almost need to redefine, let's say, for the type of images that we have, what is it that I want to extract from this particular image? I'm going to set everything for that. Okay, but if you have an image of sparse nuclei and you need to count them, you can undersample because you don't need a high resolution. So even if you undersample, it's not, it has no consequence on your interpretation of the image. So if you're not sure, you can take 5 different images, undersampled and well sampled, and you look at them and you analyze them and you see if there is a consequence of your result. If there is no consequence, it's fine to undersample. Okay, so we are all talking about what are the consequences on the analysis. So here are the conclusions of this lecture, and I'm okay. Right. So if we are assuming a perfect sample, the remove this. The resolution of the optical image created by the object is limited by the size of the point spread function, right? The way we record and digitalize this optical image can sometimes limit the final resolution in the image. And in that case, we are undersampling, and the resolution in the image is limited by the digitalization and not by the optical image. So on all systems, the undersampled details in the image are incorrect. So I'm saying it's not the case that everything in the image is incorrect. It's only what is undersampled in the image that is incorrect. So we create artifacts, lines that do not exist, incorrect intensities, and round objects that become elongated. But again, all these artifacts are only present in the objects that are undersampled in the image. So if these objects are not of interest for you, it's fine. On the single point confocal, when we acquire Z-stacks or time lapses, some signal is not recorded at all. So we have missing information, okay? Because we jump from one spot to another, we jump from one Z-stack to another, and we only record at the time point. So we may miss information if we do not sample enough. So to avoid undersampling, the distance of the XY optical resolution or the Z optical resolution must be sampled with more than 2 image pixels or Z planes. And so in XY, the image pixel size must be smaller than half of the optical XY resolution, and the Z step must be smaller than half of the Z optical resolution. On single point confocal, click the magic button. And on camera-based system, you must check that the image pixel size is also half or smaller of the optical resolution. So just look in the software at the image pixel size and calculate it. See if half, then you're fine. And on all systems, there is a magic button for Z. And in time, we need to find out what is a significant change for our scientific question, how long it takes to occur, and then we divide this by 2, and this is the way we set our interval. Half of the significant change, the time it takes for a significant change to occur, or smaller intervals. So if one undersamples anyway, the image resolution in that case is not the optical resolution, but it is the size of 2 pixels, 2 image pixels. I'm sorry about that, and I'm going to change all this and update the PDFs that I put in the lecture. So when you undersample, the image resolution is the size of 2 pixels, okay, and not the size of the optical resolution, and that is true on any system. So please reverse design your experiments. First, define your scientific question in terms of quantifiable metrics that need to be resolved. And then afterwards, you figure out what the resolution that you need for your quantifiable metrics is, and which differences you need to interpret between wild type and mutant, for example. And from this information, you detect which microscope you're going to use, which objective you're going to use, and which sampling you're going to use, which pixel size you're going to use. Okay, so we reverse design our experiments. I'm going to stop in here, and we're going